Hi everybody, it's Mr. Poller. In this video I'm going to be talking about bacteria cells or prokaryotes. These are the most ancient life forms on our planet. The oldest fossils of bacteria date to about 3.5 billion years ago. These are really different kinds of cells than plant cells or animal cells because they don't have a nucleus, they don't have organelles like mitochondria or chloroplasts. They do have ribosomes. They do have DNA that controls their genetic information. Most of the jobs that need to be done in bacteria cells are done in the cell membrane. Let's take a look at an image that allows us to compare a prokaryotic bacteria cell with a eukaryotic, this is an animal cell. We see that they share some features in common. They both have a plasma membrane, cell membrane. They both have cytoplasm. They both have DNA. The animal cell and plant cells too have a nucleus. Bacteria cells don't. They both have ribosomes because both need to make proteins. The other major difference is the size. Notice that these only go up to a size of about 10 micrometers for bacteria, whereas animal cells are between 10 and 100 micrometers. So animal cells can be as much as 10 times larger than bacteria cells. The cell envelope is the outer covering of the bacteria cells. They have a glycocalyx, which protects the cell and can help them stick to the environment where they live. The cell wall provides support and protection. That's right here in black. In bacteria, the cell wall is made of a chemical called peptidoglycan. Finally, let's talk about the cell membrane. We see that in here. The cell membrane is the part of the cell which is going to control the things that are going in or out of the cell. In this image, we'll do a quick review of the cell membrane. The cell membrane is a semi-permeable membrane. This means that some materials can pass through it, others can't. It is a bilayer. You can think of it as being like a sandwich, which is made of phospholipids. The phospho part is the head right here. This part is attracted to water. The lipid tails that you see on the inside of the sandwich, these are hydrophobic, so they tend to go into the inside of the sandwich. They are repelled by water. Other things that are included in the cell membrane, we have a lot of different kinds of proteins. There are channel proteins, there are receptor proteins, and these proteins are going to be involved in helping move things in and out of the cell, like these channel proteins. Receptor proteins can receive messages from outside of the cell which can trigger responses on the inside of the cell. The internal structures of bacterial cells include their chromosome. This is a long circular piece of DNA. This is going to be coiled around proteins. This controls the genetic information of the cell. Bacteria also have plasmids many times. These are tiny circular pieces of DNA which can be found free or sometimes they're incorporated into the chromosome. These can give cells new traits, perhaps antibiotic resistance. Also on the inside of bacteria cells we find ribosomes. These are made of rRNA, this is ribosomal RNA, and protein. Their job is to build proteins that the cell needs in order to function. The ribosomes are made of 30S and 50S subunits. S is for Svedberg units. The ribosomal genes are often sequenced to reveal evolutionary relationships of bacteria, and actually they're also used to classify different species of bacteria when they find differences in those ribosome gene sequences. There are two different categories that the prokaryotes are placed into. The eubacteria, this name literally means true bacteria. Common examples of eubacteria include things like E. coli. And then there's the archaeobacteria. These include extremophiles, which are bacterial cells that live in very extreme environments like hot springs. Now, it might be tempting to say that these are really similar to each other because they're both bacteria. The truth is that they are genetically really different from each other. In fact, the eubacteria are as different from the archaea as they are from plant cells and animal cells. So the eubacteria and archaeobacteria are actually placed into two separate domains. Again, this means that genetically they are really different groups from each other. Bacteria live almost everywhere on our planet. In fact, we haven't discovered all of them yet, and the leading microbiologists in the world can't exactly agree as to how many different types of bacteria there might be. Some place the number in the tens of thousands, others in the hundreds of thousands, others say that there could be more than millions of different types of bacteria that live on our planet. These are really, really tiny cells. Remember again, the largest bacteria cells are in the range of 10 micrometers. So we need special microscopes to see these. So we need to use compound microscopes with oil immersion lenses, and this allows the microscope to give total magnification of 1,000 times so that we can actually visualize these really, really tiny bacteria cells. Bacteria come in a lot of different kinds of shapes. There are three main shapes that we should know about. There are sphere-shaped bacteria. These are called cocci. There are also rod-shaped bacteria called bacilli. And then there are spiral-shaped bacteria called spirochetes. And very often bacteria are named based on their shape and the way that they are organized with each other. So we may have sphere-shaped bacteria that line up in chains. These are Streptococcus. So if you've ever had strep throat, it was a chain of sphere-shaped bacteria that was causing that sore throat. Here's a look at a microscope image of a really common bacteria called Staphylococcus epidermidis. Epidermidis means that this is a bacteria found on the skin, so you have this actually on your skin right now. Staph means clumps, and coccus means sphere-shaped bacteria. So we can see that there are clumps of sphere-shaped bacteria, and that's where this bacterium got its name, Staphylococcus epidermidis. It means clumps of sphere-shaped bacteria found on the skin. Let's look at some important structures found in bacteria cells. Flagella are used by bacteria for movement. Pili are used for gene exchange between different bacteria cells. Capsules can provide protection for bacteria cells, and fimbriae are used by bacteria to attach to surfaces or to attach to other bacteria cells. Here's an image of a rod-shaped bacterium, a bacillus, that has multiple flagellum attached to it. Now these are tail-like structures, and the bacteria can move these in order to propel itself in its environment. These two images show some appendages that bacteria use for attachment or mating. Fimbriae shown here. So again, we're looking at some bacillus bacterium. The fimbriae are the things that look like threads between them. So these are used for attachment. Now these might allow the, shown here, the E. coli cells to attach to each other, or it might allow them to attach to cells on the inside of the gut where they live. Here, in this image, we're looking at a pilus. This is a tube-like structure that's actually connecting one cell to another cell. And through this tube, the bacteria can actually transfer DNA. They can transfer genetic information. So it's a way for one cell that might have some interesting genes. Maybe those are genes for resistance to an antibiotic. The genes could actually be transferred from one cell to another cell. So this cell, which is receiving the new genes, maybe was not resistant to antibiotics before it got those genes from its neighbor, but once it receives those genes, now we have a second cell which is going to be resistant to the antibiotics that we use in order to try to treat bacterial infections. Here's a microscope image showing Pneumococcus bacteria. Coccus again tells us that these are sphere-shaped bacteria. Pneumo in this case is telling us that these are found in the lungs. These are actually bacteria that cause pneumonia. And what I want you to note on these images, note the dark blue spheres. Those are the bacterial cells. Now there's a lighter blue kind of outline or a halo almost around these cells. Those are capsules. And those capsules are providing additional protection for those cells, and it can actually be something which makes these cells more virulent, more able to cause disease if they have the capsule, because it protects them from our immune system that's going to try to get rid of these bacteria from living inside of our lungs and causing problems. Bacteria are really important cells. They have all kinds of important functions. If we think about their role in ecology, there are bacteria which are decomposers that are playing important roles in nutrient cycling. There are bacteria that are responsible for the processes of things like nitrogen fixation, taking atmospheric nitrogen and converting it to forms that living cells, plants, can use. Cyanobacteria are photosynthetic bacteria that produce oxygen, and they were actually the first photosynthesizers on our planet. The economic importance of bacteria is multiple. They are important for food production. Foods like yogurts, cheeses, sauerkrauts, all are produced by the process of bacterial fermentation. Pharmaceuticals, there are bacteria used to produce antibiotics or vaccines. Bacteria used for biotechnology applications, for example, the production of insulin. And then bacteria also are responsible sometimes for spoiling food, or they can be pathogens of plants, so they can reduce the amount of crops that are going to be grown. And then they also have medical importance. Now remember, there are so many bacteria that live on our planet. It's probably in the hundreds of thousands different kinds of bacteria that live on Earth. Of all of those, there's around 200 species that are human pathogens that cause disease in humans. So most bacteria are good, most bacteria don't harm us. There are, again, about 200 species that do cause problems for people, and most of the infections that people get can be treated with antibiotics. However, there are some bacteria which are developing resistance to antibiotics, so this is the challenge in medical science, to try to find new ways to treat infections of bacteria that are resistant to the antibiotics that have been successful in the past against bacterial diseases. The final thing that I'd like to discuss in this video is a really interesting theory called the endosymbiotic theory. This is actually a theory which attempts to explain the origin of the first eukaryotic cells. Those are cells that have a nucleus and have organelles like mitochondria and chloroplasts. The theory is attributed to a biologist named Lynn Margulis. Now there's some really interesting evidence that in the past there was an ancient symbiotic event where a larger host cell engulfed by endocytosis smaller cells, and they started living together, and eventually over time they became one cell. Now, some of the evidence of this theory is that chloroplasts and mitochondria actually have their own DNA, and the DNA that they have is really, really similar to DNA that's found in bacteria species that are still living on our planet today. Both chloroplasts and mitochondria are organelles that have multiple membranes, and we can understand that multiple membranes might arise by a smaller cell being engulfed by a larger cell. The genetic analysis of chloroplasts, the DNA that chloroplasts have, has been found to be very, very, very similar to the DNA that's found in cyanobacteria. These are photosynthetic bacteria. And the mtDNA, this is mitochondrial DNA, shows a very, very high degree of similarity with the DNA of a species of bacteria that causes the disease typhus. So again, this is a very, very interesting theory which attempts to explain the origin of more complex cells, eukaryotic cells, and there's a lot of evidence that does back up this idea. All right, everybody. Well, this was a quick look at bacteria, their shapes and sizes, and some of their important features. I hope you found the video helpful. Thanks for watching. Bye.